Hello everyone, welcome to Toppr again. So myself Pushpendu, I'm sure you can recognize me again. So we have been conducting these type of live classes each time. Every week we have different types of sessions. So today we are again back with the biology live class session. So in this series of sessions for biology, we were talking about the chapter neural control and coordination. So we are conducting these type of sessions not only for biology but also for different subjects. I'm sure by now you are aware of this. So I just request once more, like, keep watching us, keep asking for any doubts you have, subscribe to our channel, invite your friends so that they can also get help as you are getting help, right? So let's start with our today's session. So before that, I'll just help you recall what we have already done in this classes. So majorly in the first session, we have talked about mostly about brain. We have talked about the spinal cord. Okay. In addition to these two, so we have been talking about various types or various parts of the brain, how it is functioning, what is spinal cord, how it is formed, what is its function, what is the central nervous system. Do you remember? Like central nervous system, CNS, very important for our body. So we have talked a lot about the central nervous system. So in the previous session, we have been talking about the very important part of this chapter, which is the signaling. Okay, which is signaling. If you remember, I've been talking about the different types of neurons, the myelinated, non-myelinated also. We have talked about both. So we have talked about both these types of neurons. And very importantly, we have talked about the reflex action, or we can call it as reflex arc. Okay, so we have been talking about this reflex arc. What is this reflex arc? So just recall about, because these are the sessions, these are things we are going to require for today's session. Today's session will be quite small and easy one, so that you can, it's mostly will be recovery of what we have already started, what we have already discussed, and also today we'll add on to something. So let's see what are they. So we have talked about the reflex arc. If you remember about the sensory neurons as well as the motor neurons, correct? So the sensory neurons, they are present in our sense organs. They collect the signals collect करने के बाद they take them to the spinal cord, as well as I have pointed out there that mostly we say that this reflex arc is just involved with the spinal cord, but actually no, there are two cases. Most of the cases spinal cord actually takes up this function, but इसके साथ साथ brain भी ये function ले लेते हैं, right? Major decisions brain भी लेते हैं, but mostly we are mostly concerned with the spinal cord part when we are talking about reflex arc, correct? So in addition to that, we have talked about how a nerve impulse is transmitted through the nerve, right? हमने बात किया था कि कैसे nerve impulse transmit हो रहे थे एक nerve के अंदर से. So we were talking कि nerve के अंदर, if like this is the nerve, so इसके अंदर ही कैसे transmit हो रहा था. Like we were talking about the charges, correct? If you remember, charges across the membrane, and if there's a signal, the charges change, and in this way it is transmitted. And this we are talking about the what type of neuron? We are talking about the non-myelinated neuron, right? And now if you recall, we have discussed about the myelinated neuron also. वहाँ पे क्या हो रहा था? वहाँ पे we had the neuron in which there were myelin sheaths like this, okay? And these nodes, they were called what? Node of Ranvier. And यहाँ पे signal कैसे transmit हो रहा था? यहाँ पे transmission ऐसे हो रहा था कि वो jump करते थे, right? They used to jump from one node to the other. And we have talked about that this is 50 times faster in transmission than the non-myelinated one. So far we have talked about this. So today we'll be talking about something extra. So now, just for your recall, I'll just quickly draw the structure of the neuron once more so that it's helpful for you to just recall and understand how this works actually. So if you just remember a very rough diagram, I'm trying to draw it. So right, this is the cell body, you remember, this is the nucleus. Here we have the Nissel's granules. These are known as the Nissel's granules. Here we have what? These are called the dendrites. If you remember, what was the function of the dendrites? These were the places where the signals are received. Okay, so these dendrites, this is the cell body, or this is the actually axon. This is called axon. For non-myelinated, there is no layer of this myelin sheath, but in myelinated there we have seen this myelin sheath formed by which type of cells? They are known as the Schwann cells, correct? So this nerve, it ends somewhat like this, in which we have the nerve endings. Here we have the terminal areas of the nerve. Now we have been talking that the signal is traveling in this way, and very importantly it's always unidirectional. इसका मतलब है ये सिर्फ एक ही direction में travel कर सकते हैं, not the other way around, correct? It is unidirectional, that is from the dendrites to the terminal end of the axon, correct? Now, so we have been talking about that how the nerve impulse is traveling within the neuron. Now it's time to discuss how the nerve impulse is transmitted from one neuron to the other, correct? So let's go for it. So here I'll just show few structures. There are bulb-like structures over there. Just remember this. So there are bulb-like structures over here. Now from this place, all these places will have tremendous number of neurons. So here, if I draw a very rough diagram, there should be another neuron. Here there can be other neurons, correct? Whose dendrites are here? You understand? These dendrites are here. So these will receive the signals from this neuron. So you understand how vast it is branched. So कितना ज़्यादा branching है, इसके वजह से कोई भी signal इतना fast, fraction of seconds में we can sense all these things, correct? So let's see how this transmission is happening between the neurons. So here we'll be talking about a terminology called what? Synapse. Okay, we'll be talking about or concentrating more on the terminology synapse. Now this is very important. You should be very well versed with this terminology synapse. What is this? So if you define, this synapse is actually an area of functional contact between one neuron and the other. So you understand, this is the functional area. Functional area of contact. I hope you can see it properly. Functional area of contact between what? It's between two neurons. So functional area of contact between two neurons. So this is known as the synapse, right? So actually I'll be explaining you what is the main function of synapse, or this synapse is actually the area through which the transmission is happening between two neurons, correct? So it is also called as this is also called as axodendrite synapse, correct? So it is also known as axodendrite synapse. Now let's concentrate on this term. Why it is called axodendrite synapse? So if you just recall the structure of the neuron, let's just draw the end part. So this was something, the end part we were seeing. So we have discussed that this is the terminal end of the axon where there are certain bulb-like things. I have drawn it like कुछ bulb जैसा चीजें हैं यहाँ पे terminal and axon के, और यहाँ पे we had already shown that बहुत सारे neurons होते हैं जिसके dendrites इस जगह पे हैं, correct? We have the dendrites. So you understand axodendrite synapse. So it is called axodendrite synapse. I hope you have understood why this terminology axodendrite. Okay, so we are back again, and so now What are the different types of synapse we can talk about? Very importantly, the synapse we're talking about can be of two major types. So there are two types. One is electric synapse, we can say, and the second one is chemical. Electrical and chemical. So these are the two types. So from the name, I'm sure you can understand, आपको समझ में आ रहा होगा कि the electric synapse means that the signal is what? It's electrical. And chemical मतलब क्या है? The signal which is transmitted, वो signal जो है वो chemical signal है. Correct? So ये दो types के हो सकते हैं. अब एक synapse के structure में क्या है? So we'll talk about the structure of the synapse. Okay? So let's see what is there in the synapse. So let's draw the internal or the terminal end. So I have already shown this part. So if I just take a look on this part 1 and if I zoom it, what will you see? You'll see something like this. Can you see the bulb I was talking about? I've just highlighted it. I've zoomed into the picture and the bulb actually looks like this. Correct? So this bulb is very interesting. This bulb has certain structures called vesicles. What are they called? Vesicles. And because it is a part of synapse, they are known as synaptic vesicle. So yes, let me see if you have questions. Okay, so Shweta, you have asked what is the size of the neurons? Okay, interesting question. The size of the neurons actually varies. Okay, if I tell you that which is the largest, can you tell me which is the largest neuron in our body? If anyone knows, can you just comment it? Largest neuron in our body. Quickly. So I'll just answer you. The largest neuron is actually which is present between our knee and the toe. Okay, not the toe, the heel and the knee. So that is the area which has the largest neuron. Okay, so some neurons are quite large, some neurons are small. So it varies in length. Correct? Now let's continue with the discussion, and of course keep posting with your questions, your doubts. I'll try to solve it. Okay, now so we were talking about this vesicle. Now inside this we have certain structures. You know from where this word vesicle came? Vessels. Okay, vesicle came from the word vessels. Vessels are certain structures which can carry something. So that's the reason vesicles. Now these vesicles contain certain chemicals. So here I'll just highlight. Let's first talk about the chemical signaling first, and later on I can tell you about the electrical signals. Okay, so first let us concentrate on the electrical signal, the chemical signal, right? So first we were talking about what are the structures which are needed. So we need this bulb in which there are synaptic vesicles. So let me write it down. Synaptic vesicle. Okay, so you see these are the synaptic vesicles over here. Now very important part. What does these vesicles contain? Because I have already said these are vessels, like, so what does it contain? It should contain something. It contains called neurotransmitters. Remember this word, neurotransmitters. Okay, now from the word, I'm sure biology and science is very interesting, because from the word actually you can understand what does it want to convey to you. So neuro, something related to the neuron. Transmitters, that means it helps in the transmission. So these are the chemicals which help in the transmission process. I'm sure that something is getting clear in your head. What should be the function of these molecules, like neurotransmitters? Now, in addition to that, we were talking about what are the requirements. In addition to that, we'll have the next membrane. I'm drawing it here, you see. So we have the next membrane, which is called the presynaptic membrane. Correct? So this is the membrane. this is of which part? Of the next neuron. So this part is of the previous neuron and this part is the from the next neuron. So this is the starting of the next neuron, this is the ending of the next neuron, correct? So the signal will be transmitted from this area to here. So here there is called the presynaptic membrane. You remember this is the presynaptic membrane, okay? So also known as the presynaptic cleft. So this area is called the synaptic cleft from which the neurotransmitters will be transmitted. I'll talk about the transmission process in detail, right? So okay, so just I'll mention some neurotransmitters. A very common one is called acetylcholine or choline, right? Acetylcholine is one of the neurotransmitters. There are other neurotransmitters also like noradrenaline, right? Let me write it down. Noradrenaline. Okay, so these are some of the neurotransmitters which are present. Like acetylcholine, the most common type of neurotransmitter which is present. Noradrenaline is also present, correct? So quickly let's revise what are the things which we require. We require the synaptic cleft, the synapse, in which we have the vesicles, the synaptic vesicles. We have the neurotransmitters filled inside the synaptic vesicles. What are the examples of neurotransmitters? Remember this, acetylcholine. It can be abbreviated as various examinations abbreviate it as ACH. Okay, you remember this. You may get questions directly with the use of abbreviation. So ACH, acetylcholine, right? So we will be mostly talking about acetylcholine. So let's move on to the next part. So here what happens now? Actually, let's talk about the transmission. So I hope you can see transmission of impulse. So again, I'm telling you, keep posting your doubts. I'll of course try to solve it, and if time does not permit to answer your question right now, so but you can keep posting the doubts. We'll surely try to get back to you, right? So okay, let's talk about the transmission. The first type we'll talk about is the electrical synapse. Electric synapse. This is very interesting because you have got a bit of idea about the chemical synapse. Now what about electrical synapse? Are you confused about this? Because you see, I think you have already made out that what is happening during the case of chemical synapse. In chemical synapse, this acetylcholine, let me show you the picture once again. So we have seen this picture, right? So this acetylcholine, from which is present in this neurotransmitters, it will be released and it will carry out the signal from here to the next neuron. So it will enter, it will come out, it will burst out these neurotransmitters and they will enter in this portion. So this was what is happening here. But what about chemicals? What about, this is the chemical part. What about electrical part? How will that be transmitted? Because this is a gap, this is synaptic gap, right? So let's see what happens there. So in case of electrical synapse, what will happen is, if I draw this part, there are certain junctions actually. You see, there are certain junctions which is connecting two neurons. So can you see these junctions? So these are certain junctions which are connecting the adjacent neurons. What are they called? They are called as the gap junctions. Now, so let me see if there are questions. Okay, so Shweta, you are asking that there are many transmitters present in our body. Yes, we have different types of transmitters. Like I've already mentioned about two important, like noradrenaline, we have the acetylcholine. There are many different types of transmitters, but in most of the cases, this is actually if you study more detailed about molecular signaling, you can get a very good idea about this. But actually at this point of time, you do not need to know about molecular signaling. But yes, there are many different types of neurotransmitters. So okay, let's get back to the discussion. Gap junction. So these are called the gap junction. Now the question is, are these gap junctions part of the neuron? What do you think? Actually, saying these are not the neurons, because these are nothing but made up of proteins. So these are proteins, understand? So there are certain proteins which actually help in the transmission. These are called the tubular proteins. Tubular proteins. Okay. 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 So Anupam, you are asking what is the ACHO? That is acetylcholine source. Okay. So here only, like, let me show in this picture only. So there are certain areas, so which this acetylcholine, they are produced back here only. I'll just discuss about this. The neurotransmitters, they are released. I'll just highlight in very brief. The neurotransmitters are released, and you'll see these vesicles are again revived back. Okay, when there is hydrolysis of this acetylcholine, and it actually results in the formation of different materials, and they get reabsorbed and they recombine and form acetylcholine again. They come back into this previous neuron. Okay, we'll be discussing about this. So okay, so these are called the tubular proteins. So from the name you understand, these are tubular. So these act as nothing but channels. These are channels. ये channel जैसे काम करते हैं और tubes जैसे काम करते हैं, जिससे इलेक्ट्रिकल सिग्नल सिमिलर वे जैसे हमने डिस्कस किया है ऑलरेडी, जो एक्सॉन में ट्रांसमिट हो रहा है, उसी तरीके से प्रोटीन ट्यूबुलर प्रोटीन्स के अंदर से भी ये ट्रांसमिट हो सकता है. दीज आर चार्ज प्रोटीन्स. ओके, सो दिस इज व्हाट इज हैपनिंग इन केस ऑफ electrical synapse. So these are the gap junction. This is, you know, this is the synaptic cleft. There is certain fluids present over here, and these are the proteins. Correct? Now, can we talk about in detail the second one, which is the chemical, right? So now, let me quickly draw the second part. So these are the vesicles. If you remember, this is the next presynaptic membrane. Correct? Now what happens? So here, slowly and steadily, let's see the steps which is happening, because this is the important part which we need to answer. And in fact, chemical signaling is much prominent in our body, but you understand electrical signals are much faster. Because let me explain this. I think you can understand yourself why this electrical signal is faster. So the electrical signal appears from this area, from the neuron, from the axon to this portion. Due to this change in electrical potential of this area, these vesicles, they what happens, there are calcium ions which are secreted by this. Okay, these areas, calcium ions are present over there. So these calcium ions, actually what they do is they push these vesicles towards the periphery. So they come here, they combine with this portion, this membrane, and they release, they burst and release. Okay, so what happens, they release these neurotransmitters. You understand? They release the neurotransmitters. So the calcium ion causes the movement of the synaptic vesicles to the surface of the knob. So this is the knob portion, you know. So here they move to the surface and their contents are discharged. Now what happens? So here these portions have certain protein receptors, because you understand these are the chemicals, the acetylcholine. Let's take an example of the acetylcholine. They are released, and this causes a signal, and which is received by the protein structures or the protein receptors present in the presynaptic membrane. Okay, because of this signaling, the protein conformation changes, which brings about a change in the membrane potential and the electrical potential of the membrane. So you understand? Let's see. So the electrical, this was from till this portion, it was electrical impulse, which was changed to chemical signaling. So these chemicals were released over here. Again, the chemicals were accepted. The chemical signals got transmitted to electrical. So here it was electrical, here it was chemical, again transmitted to electrical, and then it will transmit to the next neuron. And the same thing is going to happen all throughout the reflex arc. Correct? So I have said you the synaptic vesicles, they will return over here, correct, to the cytoplasm of the synaptic knob. They will return, the vesicles will be formed, and they are refilled with the transmitters, neurotransmitters. So you understand, when the signal comes over here, it causes the depolarization. If you just remember this terminology, depolarization, right? Depolarization, then repolarization happens and the signaling is continued. ऐसे ही signal pass होते रहते हैं, right? Depolarization, repolarization and then resting potential. आपको अगर याद होगा तो इस तरीके से it keeps on traveling. So it causes the depolarization and it generates the action potential, right? These are the terminologies we have already discussed in a previous session, so you must be remembering this. So depolarization causes the action potential, generation of the action potential. So from the resting potential it moves to the action potential, again it revives back to the resting potential, right? So if you remember about the refractory phase, right? Now, so I think we got a question from Anupam, right? You were asking how the acetylcholine is regained back. Now what happens, I'll say, the acetylcholine when it is released, there is hydrolysis of this acetylcholine, which results into the formation of acetic acid and choline. Okay, so these are the particles which can be reabsorbed again. So you understand, the neurotransmitters are released, it causes a signal over here. After that, hydrolytic enzymes are present in this area. So they cause hydrolysis, which results in the formation of two products, acetic acid and choline. Acetic acid and choline, they are reabsorbed by this knob and it is again refilled into the into these vesicles and it regains back the neurotransmitters. So you understand, this is a cyclic process. So new neurotransmitters need not be generated. Correct? So here, this is the normal thing which is happening over here. Now we have talked about different types of neurotransmitters, like I can jot down something, acetylcholine, okay, noradrenaline. So these are certain neurotransmitters we have been talking about. There is another one, so norepinephrine, okay. So Shweta, you are asking about the different types of neurotransmitters. Norepinephrine is there, there is gamma aminobutyric acid, okay. Then we have also talked about noradrenaline. So there are various types of neurotransmitters, but acetylcholine, norepinephrine, these are the major types of, or they constitute the major parts, correct? So we have been talking about these various transmission of the impulse. We have talked about two major transmission, that is electric synapse and the chemical synapse, okay. So let's quickly highlight the difference between this. So here, now we were talking about what happened in electrical synapse, what happened in chemical synapse. Now I hope you can understand in which way it should be faster. It should be faster through the electrical synapse. Why is this so? This is so because you understand, there is a junction over here. It's simply a connection, so it is linked, okay. It's a simple connection. But in chemical, there is no connection, there is a gap over here, and this connectivity is done through the neurotransmitters. So it will take time, obviously, because this is the area in which the charge will simply pass through and it can be transmitted, right? So this was whole about how the transmission of nerve impulse is happening, or the synaptic cleft or the synapse. So this is a very important area in which students get very confused about what is synapse, what is the synaptic cleft, what are the different types of transmissions. So I think in today's session we have covered that main part of this discussion. So keep posting your questions. Let me see what questions you have. So we are saying is gap junction is present in every neuron? Then what about synapse? Okay, no. So that's what we are telling. This gap junction that I'm talking about, they're present in certain types of neurons, okay, not all. That's the reason we have the chemical signaling which is present in certain other types of neurons, okay. So some of the areas, they take care about this transmission is taken care by chemical signaling, some areas in which it takes place by electrical signaling, okay. So let me see, if you have other questions, keep asking so that I can answer your questions quickly. Okay, so you're asking, Okay, Asdul, you asking what is neurotransmitter? Okay, fine, I can just tell you. Neurotransmitter, those are nothing but chemicals. Just remember it, they are chemical molecules which help in signaling. So the neurotransmitters, these are molecules which were present over here. So why are they important? Neurotransmitter is nothing but they have the capability to take the electrical signals and get converted to this chemical signal. So electrical signal is converted to the chemical signal through the neurotransmitters, and this neurotransmitter help in again conversion back to the electrical signal in the next neuron, right? So here it's nothing but because of the charge present in these molecules, these particles, it imparts a charge to these receptor proteins. You understand? So it's nothing about nothing complicated, but it's all about charge and the potential and the chemical gradient. Okay, everywhere all this signaling takes place because of two factors. There's a gradient that has to occur, either electrical or chemical. So in this case, the same thing is happening. Okay, so let's see if you have other questions. Okay, so yeah, we have got a question like what is the role of calcium here? Okay, so I've already mentioned like calcium, it helps in the movement of these vessels, these neurotransmitter filled vessels towards the periphery. Okay, and also helps in the bursting. You know what will happen? It is called, there are certain techniques which is used in bacteria also called pinocytosis or exocytosis, right? So these are certain, this is actually exocytosis, in which it will combine to the membrane and become a part of the membrane and open it up. So you understand? So if this is the membrane, this thing will come, it will combine and it will help in opening it up. So this is the way in which neurotransmitters are released. Okay. Okay, so again we have a question. Prathamesh, you are asking what is the difference between electrical and chemical synapse? Okay, I think we have discussed already, but I'll quickly highlight you. So electrical signal and chemical signal or chemical synapse, the difference is the transmission. Here it is happening through electrical potential. Here it is happening through chemical potential. Here we do not actually have the gap junction. Here we do not have the gap junction. Okay, so here it is connected by the protein membranes or the tubular proteins. Here we just have the vesicles which releases the neurotransmitters and it is connected back. So I hope you have understood what's the difference. Main difference is basically the transmission is happening based on chemicals. Here the main transmission is happening is based on the electrical signals. So let me see if you have other questions. Okay, so I think for today we are done with this part of the section. So again, I'm telling you, like, keep subscribing our channel. We have a lot of sessions which is coming up every day, and keep visiting us. So invite your friends so that they can also get helped by us. So this is what we have covered in today's session. So we'll be continuing these classes soon. So if you have any doubts, keep posting us. We'll try to get back to you as soon as possible, right? So thank you for joining us. Thanks a lot.